It is also important to understand, to have a clear idea of how it is that vision works, that there is a particular, like the wavelength is going to determine, again, the hue or the color we perceive from any object that we observe in our environment. But it's also important to understand that we cannot, as humans, we cannot perceive every single type of wavelength. In fact, there is a huge spectrum that you can see right here in this upper line right here. There are all these cosmic rays, gamma rays, X-rays, ultraviolet are all types of electromagnetic energy that are different from one each other depending on their wavelength, and we can only capture or perceive a very tiny bit. This part that is colored here in the middle, we can only capture that tiny little bit of the entire electromagnetic spectrum. And this is, and usually it's basically very, very small, the amount of the amount of color or different wavelengths that we can capture and perceive. These are measured and go from 400 nanometers all the way to 700 nanometers. A wavelength, electromagnetic energy that is input, that has a wavelength measured in 400 nanometers is usually a violet bluish type of color, and when it has when wavelengths are 700 nanometers, which is close to the maximum we can perceive, anything beyond that we cannot, that is basically our threshold for sight, that we can perceive. It's usually a reddish color, and again, this is at the threshold of 700 nanometers. But anything beyond that, or anything below 400 nanometers, is beyond our thresholds, and therefore we cannot perceive it. There are other animals that can perhaps perceive something lower than 400 nanometers, or above it. If you remember from grade school, the spectrum of visible light, and that's an important term we should know right here. Visible light, when we refer to the spectrum of visible light, we're referring to the spectrum of the type of electromagnetic energy that we can perceive through our sight. Humans, referring to, when I say we, I'm referring to humans. So visible light is that it's falls into that very tiny spectrum that is visible for humans, and the gamma of colors that we can perceive. If you remember from grade school, the abbreviation Roy B Give is an easy way to understand the variations of wavelengths that lead us to see different hues in a spectrum of visible light, and it goes from red, orange, yellow, blue. As you might see, it's basically in reverse. We have red right here, going all the way to red, orange, yellow, blue, green, I think it's indigo, and violet. And these are the colors. Any color we perceive is a combination or falls in that spectrum, going all the way from 700 nanometers, red, to violet in 400 nanometers. And just to make sure you understand, it is these vary, this spectrum varies in terms of their wavelengths. As you saw in the previous slide, longer wavelengths, 700 nanometers, lead to a reddish color, and those, when the crests of the waves are closer to each other, that means that it would be closer to, it's a smaller wavelength, measured in nanometers, closer to 400, and that would lead to a violet bluish color. So now that we have a better idea of how electromagnetic energy is input in our brains, it begins in the form of light, of electromagnetic energy, in the form of waves. We discussed how light basically operates, and now it's imperative to understand the gross anatomy of the eye and the functions of its main parts, at least as they are pertinent to this unit. So let's start with the white overcoating of the eye, which cannot be appreciated pretty much in this diagram, but it would include this area right here. And this white area is called the sclera. I hope I'm typing it correctly. Something tells me I'm not, but I'll check it if it's with a CH or not. But that's the white part of the eye, and it's important to understand because although its function is not that decisive in the process of vision, it provides eye with its form, its shape, that round or orb shape it has, sorry, and it helps to protect the eye too. The eye, remember, is filled with fluid, and this fluid presses outward. So the sclera gives this fluid. The sclera is basically giving this fluid something to press against. So it gives the eye its shape. Then we go on to the next part of the brain, which is the cornea, located at the very front of the eye, and it's clear, transparent, and it's this curved membrane, and it covers and protects the pupil and the iris. We're going to talk about those in a minute. And the cornea, so the cornea protects the iris and the pupil. That's one function, and the other is that the cornea focuses light on the retina. We're going to understand these other parts in a minute. Again, I'd like to repeat that the cornea is sort of the clear part of the eye where you put your contact lenses. For people who have worn contact lenses or wear them now, wherever you put them, that's your cornea. It's also, I'm pretty sure it's one of the two most sensitive parts of the entire human body, the other one being the eardrum. So any type of pain in these areas is going to be, we're highly sensitive in these areas, so you don't want to mess with your cornea. Very, very painful. In the middle of the cornea, behind and in the middle of the cornea, is the pupil. And the pupil is that black circle inside the eye, and it's basically nothing, meaning it's nothing with a black surface. It's just empty. And the pupil exists basically because, yeah, there's nothing there. It's really an opening in the center of the iris, which is the other part we need to talk about. And the iris is a ring of muscle. That's important to understand. That controls the amount of light entering the eye, and so the pupil gets smaller, meaning that or larger, depending on how much light is in the environment. And the iris is the monitor of this amount of light and says, okay, I'll make the pupil smaller. So remember, the iris is a muscle, so it'll contract, making the pupil smaller, controlling this area, this empty area or this hole, if we are in a bright environment, and so less light will come in. But in a dim environment where there's not much light, the iris then says, okay, let me expand, and then I will allow more light to come in, and therefore the pupil will enlarge or expand as well. And again, the reason why the pupil is black is simply because there is no light inside of our eyes. That's important to understand. And it's important to understand because when we see, if people had light in their eyes, if we had light in our eyes, then what we would see in other people, and it would be very strange, it would seem like, I don't know, something very strange, is this light beaming outwards and our pupil shining out, which would be kind of scary, if you ask me, sort of zombie-like. But the thing is that we have actually seen this, and it's often with photography. So I'm sure we have taken a picture, or we have appeared in a picture, and we show up with red eye. And this is basically this illustrates how it would look like if we had a light in our eyes, because what basically is happening when we see ourselves or others with red eye in a picture is because the light of the flash usually enters the eye and bounces the back off that surface, so off the light sensitive retina that is in the back of the eye, and it flashes back towards the camera. So that is what we would look like, which is not a very pleasant image, if we had light in our eye. And this, again, just to remember, this is an example that illustrates why our pupils are black, because there is no eye, there is no light in our eye, basically. So going back to the iris is that colored part of the eye, and that's the part that people comment on when people say, "Oh, you have such pretty eyes, you have beautiful eyes," whatever the case might be. And again, I'd like to reiterate that the iris works hand in hand with its companion, the pupil, and it's sort of like a donut. And the donut itself would be the iris, and the hole in the donut would be the pupil, or the pupil would be the what do you call those Dunkin' Donut things? I don't know. I forget right now. Having a brain fart right now, but you know what I'm talking about. That would be the pupil. Then let's move on to the lens. We're going from forward from the cornea, the most forward part of the eye, going entering its structure, and the lens is located behind the pupil and iris. And a lens, it fine tunes the focus of the image that we look at. Sort of by, and it's important to understand that the lens is located in this liquid, and it gives itself some saline solution, I believe, that it sort of gives itself, this property of being in this solution, gives it some flexibility. So what the lens does is, when it's focusing that image, when we see something blurry and we're trying to focus our vision, what we're doing is flexing the curvature. And this process, it's important to know, is called, made by the lens, is called accommodation. And it's not only important to understand this process for this unit, but it's also important to differentiate it from schema accommodation. So in a camera, now in our eyes, we only have one lens per eye, right? So it needs to be flexible. Now in a camera, we have several lenses that operate the same way, but there, with a camera, we can choose. If we're talking about a professional camera, mostly, so we can choose from different types of lenses based on the distance between where we are and the object. But with our eye, we only have one, so we have to work with this imperfect but pretty flexible one that is going to adjust its curvature, right, how it's situated, in order to focus on the object that is the what we're seeing, or what we want to focus. It's important to mention that this curvature, the flexibility that our single lens has per eye, works close-handedly with the iris. And what it does is, so you picture that ring, the iris is a ring of muscle, sort of pulls on this lens, changing its curvature of that lens, and it is this process that allows us to focus, whether it is in bright light, in dim light, or any variations regarding light conditions. So let's say you go into a dark basement, right? And your lens needs to accommodate under these situations where there's not much light or no light at all. And so what it does is the iris helps. Iris helps whether to pull this because remember, the iris is a ring of muscle, so it'll pull this lens, and the lens will be changing its curvature, helping us accommodate, and therefore help us see better in. And it usually is very quick. It usually happens within a minute. Now, you will see best. The process of accommodation can take a while, and it has been measured that I think we can see best. Let's say we walk from a bright light conditions into a very dark conditions in a basement, as in this case, and we will see in less than a minute we're already going to start this accommodation process, and therefore adapt to the environment. But we will see best, this process continues, and we will see best in about 20 minutes. So have those two time frames in mind. And the process of accommodation is not that smooth sometimes. And remember, this sort of illustrates the next part of the eye that I'm going to talk about, which is the retina. But the retina is very light sensitive, and let's say you've been sleeping and you need to go to the bathroom in the middle of the night, in the middle of the night, and you go to the bathroom and you turn on the lights. Of course, in this case, accommodation is occurring too fast, and because our retina is too sensitive to light, then it sort of hurts. But basically, what is occurring is a process of accommodation too. The lens is saying, "Oh, now I have a lot of light all of a sudden, and it needs to change its curvature." And this is basically what is causing the pain by too much light entering and striking our retina, very light sensitive retina, and the iris is sort of again pulling or changing the form of the curvature of the lens, and all this is happening in a fraction of a second, and therefore it can be painful. But this is again for you to remember that the process of accommodation needs to occur, and it's helping us adapt to the environment, even in such sudden cases as it is when we go from being asleep, and then we, in this case, as an example, you go to the bathroom, you turn on the lights, and all of a sudden you feel that it's painful. The accommodation process could be not so smooth as with the previous example. Okay, so the retina is very, very important, and it is this area right here. It is the light sensitive surface at the very back of the eye, and it houses rods and cones. We're going to talk about those in a minute, but these are the photoreceptor or light sensitive cells. And the retina is very specialized tissue. In fact, it has sort of like its own nervous system, because it has grown actually from the same. It's basically, let's just say, it's basically an extension of that same tissue that we have in our cortex, and it specializes in light reception. So the retina is the area in the back of it's the tissue in the back of the eye where the image is projected upside down, in fact. We'll later talk about how we invert this instead of seeing the world upside down. But for now, understand that the retina is that tissue where the image is projected. And for now, I'd like you to, before we go on to the next portion of the eye, I'd like you to look around and take a minute to glance around you, and you'll notice that it seems that you can take everything within your visual field. If you pay attention, however, you can only see one thing clearly. So stop glancing around and now focus on one object wherever you are. Pick any object, and then focus on one object, and you'll notice that you can see that object very clearly. Of course, for those who wear glasses, hopefully you have them on, or picture that you can focus on that object. The point here being is that you can see this object that you're focusing on, that you're focusing your selective attention on, and your visual field. You can see one image very clearly. And the reason why you can see that image in a much clearer way, what I mean by clear is that you can define it, you can notice its surface in detail, notice its dimensions, etc. And the reason why you can focus on it, why you notice all the details of that object, is because that object that you're focusing on, that is in, let's say, the center of your visual field, is being projected in your fovea. The fovea is the central area of the retina, and it's where our vision is most clear. The fovea consists solely of cones. Remember, I said the retina contains rods and cones. The retina contains rods and cones, and the fovea, let's call it the VIP center of the retina, where whatever image is focused on this part of the retina is what we're going to see more clearly. And the reason for this, the reason for why we can notice the details, the colors in more detail than anything in our peripheral area of the visual field, it's because it has its great, it's because this is the VIP section of the retina, as I said previously, and it's the area where, it's the area of the retina that has the greatest density of photoreceptors over any other spots on the retina. And this area too consists solely of cones instead of rods. So just to reiterate, the fovea is the prime time spot of the retina, and it is here where we can experience exquisite vision. The reason for this being that it contains cones, and it's again the center spot where we focus our vision to. Now, the opposite of the fovea is the sort of the ugly duckling of the eye, is an area where we do not experience any vision at all, and this is called the blind spot. The blind spot, and when I talk about right here, in this area right here, the blind spot, and when we talk about blind spot, we're not talking about the blind spot that we refer to when we are driving a car, for instance, in which, you know, the car is riding right to the side of you, but it's not quite in the side of you and in your rear view mirror, neither it is in one of your side mirrors. But it is similar, and this parallel example can help illustrate what the blind spot is. But it is, more technically, it's that area, it's that one area that we cannot see, basically. The interesting part is that it's difficult to experience the function, or lack thereof, of the human blind spot, because our brain actually fills in the gaps or compensates for this blind spot with information taken from our expectations, past experiences, memories. So again, we are not experienced vision, but we do not know, we're not consciously aware of our blind spot on a daily basis because our brain is basically filling in those gaps. Basically what our brain does is, okay, this is in my visual field, and I am going to fill this, I'm going to fill in the blanks, and therefore that's the reason why we are not constantly noticing, oh, I'm not seeing this because it's falling, this object is falling within my blind spot. But for now, notice that the blind spot is the area, the reason why we can't see, it's because the retina at a certain point needs to send this information to the visual cortex in our occipital lobes. And this is sort of the exit of this area in order to send the information, the neural signals, to the through the optic nerve. So again, let me repeat that. The blind spot is located in an area where we cannot experience vision because it's this. See it as a hole through which the retina is sending the information, the neural signals, to the visual cortex. So it needs to escape the eye, let's put it that way. And for this reason, this area does not have cones or rods, and therefore, because it's an area, specific area, where the neural signals exit. So see it as a sort of an open door, and for this reason, we cannot experience vision in this blind spot. So just to leave it very clear, the blind spot is located in the retinal area, where without receptor cells, that means without any rods or cones preventing us from seeing anything. Now, if you'd like to experience the blind spot, let's go on to the following exercise. To find the blind spot of your left eye, look at the image in front of you with both eyes open. Now, keeping the monitor at sort of an arm's length in front of you, close your right eye and stare at the circle with your left eye. Now keep staring at the circle, at the dot, while you move your head slowly towards the monitor. The cross or the plus sign located on your left side should disappear or mostly disappear. And the reason for this, when you have found that plus sign has disappeared, it should be somewhere between 6 or 12 inches from your eye. The reason why is because at that moment, at that distance, that plus sign is falling under that blind spot. Now, again, we don't usually notice this because our eyes are constantly moving and our brain compensates by filling in this blind spot by using information taken from our past experiences, memories, and expectations. But again, this exercise is to be consciously aware that there is a blind spot located in the retina area where the retina is sending the neural signals to the visual cortex through the optic nerve. It's where the optic nerve exits the eye in order to communicate with the occipital lobe and its visual cortex. So let's explain now the rods and the cones. So if you remember, I mentioned earlier that throughout the retina we'll find these rods and cones which are basically our photoreceptors or receptor cells, visual receptor cells. They take in the light waves from the retina. Now, they are called rods and cones, so you can see here in the expansion, they're located in this area right here. They're called rods and cones because of their shapes. The cones are more, both of them are cylindrical, but the cones, as you might see, those red and blue cones that you can see in this area, are cylindrical, but they have these cone pointy topish type of things, and the rods, well, they are still cylindrical, but they have no pointy. They don't have this pointy top. So the rods are responsible for dim light and color vision. Another important difference is the location of the rods and cones in our retina. As we said, right above the blind spot, we can find this area. It's sort of a pit, and it is thanks to this pit that cones. That pit is called the fovea, as we mentioned earlier. And because of this pit, it's a perfect location for cones to receive light directly. And it's important because they do require a lot of amount of light. Remember, they help us see during bright light conditions. And that pit is only, that fovea, it only houses cones. And this is why when we focus on an object, we are able to appreciate its colors, its surface, its definitions in a clear sense because of the location, and because this location, the fovea, houses only cones, allowing us to see, appreciate its colors, because they do process color, whereas rods located in the peripheral area of the retina can only, they do not process color, but require less light to react chemically. Therefore, when we are in a situation and this is the reason why our peripheral vision, believe it or not, does not see color as well defined as we do when we focus the image in the rods. You can test this by focusing for a moment again on one object that is in front of you, and then try to notice how your peripheral vision, try to focus on that, or the quality of color and definition of the objects that are not located in front of you, because you are not focusing them in the fovea, in this prime time area of the retina. Because the objects in your peripheral vision are not focused on this, you cannot clearly define these colors. In fact, if you would like to take this up a notch more formally, you could try the following exercise. I mean, grab several crayons or markers if you have, and then look towards the front of you, and pick, without knowing the color that you pick of those markers, and put the arm sort of at a 90-degree angle, and sort of put it away from you, your arm, with that marker, and try to see if you can guess the color, if you can appreciate the color of that marker that you grabbed without knowing. Can you see if it's a blue marker or a yellow marker? The likelihood is that you cannot, because again, the marker is not in front of you, it's not being focused, this image is not being projected in the fovea, that has all these cones that would help you appreciate the color. Instead, you are seeing this marker, trying to see it with your peripheral vision, and your peripheral vision is not being focused on the fovea, instead on the peripheral area of the retina that contains more rods. And again, these cannot help you discriminate the color of that marker that you are trying to find out the color of. So to briefly summarize these photoreceptors, the two types of photoreceptors we have, we can look at the following chart, and this chart is very important to know if you're asked to compare or contrast them. And as you can see, rods, we have 120 million of them in each eye. We have many more. Remember the ratio, 20-1 approximately, whereas we only have 6 million of cones in each eye. So by number, you can see a significant contrast. Rods help us see in dim light conditions, whereas cones allow us to see in bright light conditions. Rods are responsible for achromatic vision, chroma, color, achromatic without color, and cones allow us to see chromatic vision. Rods provide us with low visual acuity, try to recognize that term, whereas cones allow us to see high visual acuity, and basically what this means is acuity is the degree to which we can define, see the details, notice the colors, and in fact the difference of colors of one single object. And one other difference that I'm surprised we don't find it in this chart is that rods are located in the periphery of the retina, whereas the cones, we locate them in the fovea, the area of that pit that harbors only cones, and where the light waves go to directly, unimpededly, and therefore light waves can be processed quicker and more efficiently. But don't think that rods, just because they provide us with achromatic vision, that is, vision without color, are not as important as cones. If we are, I'm sure all of us have experienced that we are in let's say we are in a dim light situation where we experience very little light, and but after a certain time, we are able to see more and more. I think I mentioned it previously, but by less than a minute, we can already see that our vision conditions are improving, and at 20 minutes, we can actually see a lot in the dark. And this is something called, for which we thank our rods for, this is called dark adaptation, where we get used to these conditions, and because rods don't require much light waves in order to react chemically, they are helping us navigate through dark conditions. And again, this process that takes, it's actually quite quick and fast. By 1 minute, we notice that conditions in dark, dim light are improving, and by 20 minutes, we can contrast and be quite surprised at how much we can see in the dark thanks to our rods. Again, dark adaptation, we should thank our rods for that.